Okay, so welcome to this class. In the last class we have gone through briefly about the composition profiles, 1D composition profiles, 2-dimensional composition profiles, and also we described the proxygrams, which means that proximity histograms, where we can, if you have some interface which is curved and which is not flat, then you can actually accurately determine the composition profile across that particular interface by using the proximity histograms, correct. And the last two classes we also described about the sample prep by focused ion beam, and I suppose you have gone through those videos where you have seen the step by step process of making needle specimens for the atom probe. Fine. So in this class what we will do is we will talk about some more data analysis, what type of information you will get it from the reconstructions, correct. So now what we call these are the frequency distribution analysis. Okay. So what is this frequency distribution analysis? So usually these are helpful in cluster analysis. I'll come to these terms in later in the then decomposition effects in the microstructure that may be related to the phase separation and formation of precipitates. Decomposition effects, and also the these two we can get the atomic scale quantification. Atomic scale quantification. Fine. So okay, and so the first part which is described in the frequency distribution analysis is, we call it is a binomial analysis. This binomial analysis, so this is nothing but a grid based frequency distribution. Okay, so this is a grid based frequency distribution technique. Okay, so here what we do is each voxel, I think the term voxel has been explained or described during I think the last few in the last few classes. So each voxel usually here contains a constant total number of atoms, which is given by NB, correct. So what is the so this is the each voxel. So how the analysis is done? So for the analysis it is means that the frequency with which atoms of some particular element, for example A, atoms of some particular element A occur per block. Okay, and this is compared to the theoretical distribution of A atoms that are present or occurred by random in the system. Okay, throughout the data set, randomly throughout the data set. Okay, so it is just to determine the frequency with which the particular element A, which is present per block or per voxel, what we can per block, as compared to the A atoms which are randomly distributed throughout the data set. So this is the comparison. Okay, and this random distribution is nothing but the binomial probability distribution, okay, which is meant by PB. Okay, so this random distribution of A atoms throughout the data set, it is nothing but the binomial probability distribution, which is PB. So if there is a material, or if there is an alloy with the proportion of A, which is XA, correct, then the corresponding binomial distribution means the corresponding binomial distribution can be given by F B N, which is equals to N PB, and which is equals to N NB factorial. This is a mathematical term, there is no need to remember, but it is just to understand what is this binomial distribution. Factorial XA N 1 minus XA NB minus NA. Okay. So this is the term for the binomial distribution of A, which is present as the proportion or the fraction which is XA in the sample or in the data set. Then the binomial distribution of this particular species A can be given by this formula. Fine. So if the construction, if there is a reconstruction, which is divided into n blocks, I will give you the example for this, which is divided into the n blocks. Okay. Now the FBN is the is expected that expected is the number of FBN is the expected number of blocks each containing n atoms of element A. Okay, so this particular function FBN can be plotted in comparison to the experimental frequency distribution, which is En. Okay, so this FBN is nothing but the expected number of blocks of each containing n atoms of element A, correct, and this can be plotted in the comparison of experimental frequency distribution. Okay, so this experimental frequency distribution, whatever you are getting from the needles from the reconstruction, usually have a shift away from the binomial distribution. Okay, if there is a shift as compared to the binomial distribution of this particular element A, it indicates that the atoms are arranged. The atoms are arranged in a non-random manner. Okay, so if these atoms are arranged in a non-random manner, it means that the A atoms, the A elements, or what we call it might be possible that there must be a multi-phase which are presented, or it might be the atomic scale clustering or nanoscale clustering, atomic scale or nanoscale clustering existence. Okay, so this binomial distribution is for the random arrangement of A element within the data set. Okay, this is given by this. And if you plot experimental frequency distribution En with respect to the binomial distribution, then if there is a shift in the on the peak, or there is a shift in the frequency distribution, it shows that the atoms are arranged in a non-random fashion. Okay, this hints us that this might be presence of multi-phase alloys, or there might be nanoscale clustering in the alloy. Fine. So I will show you an example of atomic scale clustering in a aluminum copper alloy. Okay, so here you can see that the orange color are the copper atoms in an aluminum alloy, and this particular A is as quenched, means it's a solid solution, mostly it's a solid solution. Then these are heat treated at a certain temperature and time, so maybe you can put it as a T1, 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 T2. Okay. Now here in this, each data is divided into equal blocks of 200 atoms NB. Okay, and these are contained same number of blocks. Okay, so each data set is divided into equal blocks of 200 atoms. It means each block contains 200 atoms, and each data set has the same number of blocks in the data set. Okay, so in as quenched, and you can see there is a plot between the counts and between the copper atoms per 1000 atom block. Okay, so this x-axis gives usually it gives the concentration or the number of copper atoms per block, number of copper atoms per block. And these are the counts. Fine. So if you see this black color curve, which is as quenched, the black color curve, and as compared to the binomial curve. Binomial curve, as I told you, binomial distribution, it is nothing but the random distribution of copper atoms in the data set. So if you see that this dashed line is the binomial distribution, and it was then there is a data set 1, data set 2, data set 3, and for all the 3 data sets these frequency distributions has been plotted. Okay, so the black corresponds to as quenched, means if you heat treat the sample, it will be in the solid solution state and it has been quenched. Correct. So you can see that in as quenched sample, if you see the atom map, you can see that there is no spatial correlation. But if you plot the frequency distribution, this particular frequency distribution, the black color curve, you can see that the experimental distribution, the black curve gets widened and it is shifted to the left. Okay, so you can see that this particular the black color curve, which is as quenched, as comparison to the binomial distribution, you can see that it got shifted to the left and also it got widened. Okay, and now you can see that the after thermal treatment, like at certain temperature T1 and T2, okay, you can see that the deviation increases. T1, T2, the deviation increases and the peak shifts left more and more as the time increases. Okay, it means that it goes towards the lower copper concentration, As I told you, in the x-axis, these are copper atoms per 1000 block. You can see that as your time progresses, it goes towards the peak shift towards left. It means that this peak appears at a lower copper concentration. What it indicates? It indicates more blocks contain fewer copper atoms. You can see that these are the counts, means the number of blocks. So this particular, after thermal treatment, you can see that the more number of blocks contains fewer copper atoms. And the second is, you can see that there is a long tail as compared to the as quenched sample or the binomial distribution. So there appears a long tail. It means that long tail, it means that smaller number of blocks contains high copper concentration. Correct? You can see that in these regions there is a large tail that shows that the copper concentration is higher at these. So the very smaller number of blocks, around less than 500 number of blocks, contains higher copper concentration. So this particular analysis shows that it reveals the copper segregation effects. Copper segregation effects in the data set. You can see that these are the copper rich regions. Okay, so this we can quantify by the frequency size distribution, frequency distribution. Fine. Okay, now with introduction of these frequency size distribution or frequency distribution plots, what we can do is actually we can get the atomic distribution in the data sets. Okay, so atom probe tomography or in the atom probe reconstruction, this is a very powerful tool where actually you can get the atomic interaction distances, okay, in 3 dimension. It means that you can actually in 3 dimensions you can get these atomic interaction distances. Okay, so atomic interaction distance, these are nothing but atom to atom distance distribution. Atom to atom distribution. Fine. So with this atomic interaction distances or atom to atom distance distribution in 3 dimension, what we call it is the first is nearest neighbor distribution, which we call as a 1NN, nearest neighbor distribution, 1NND. Correct? So this is the most fundamental study where you can get the atomic distance with the APT reconstruction. Okay, so this is the nearest first. So 1 is corresponds to first nearest neighbor distribution. Okay, so this technique is nothing but the inter-atomic spacing or interatomic separation in the immediate vicinity of around each atom. So it measures the interatomic spacing of the first nearest neighbor of each solute atom. Correct? So we can do analysis by the nearest neighbor distribution by including all the atoms, or you can limit the distances between particular atoms. Okay, so it might be distance between AA or distance between BB type of atoms. So you can actually take either the analysis between all the atoms, interatomic distances, or you can limit distance between the particular atom. If there is a A element, you can actually get the distribution, nearest neighbor distribution of A to A or B to B. Okay, and these distributions are generated to analyze the distance separating each atom and the closest nearest neighbor. Okay, so it generates the distribution, distribution which to analyze the distances separating the each atom with the closest nearest neighbor of that particular atom. Okay, so here I am showing you an example. Okay, so you can see that this particular diagram, you can see that this is the distribution of first nearest neighbor as compared to the first nearest neighbor with random distribution. Okay, so this is computed by the binomial distribution function. Okay, so and this is the experimental one, which is actually present in the data set. First nearest neighbor means these are the distributions. Consider that this particular red, so there are two kinds of atoms. One is red, and one is blue. So blue is the blue number of atoms on matrix, or the blue atoms. Okay, and the red color atoms, assume that red are the tin and blue are copper. Fine, and you can see that this red color atom, if you draw a nearest neighbor, the first nearest neighbor, then you can see the distance between this particular this atom and this first nearest neighbor atom. Okay, and similarly for each atom it is calculated. For example, if you take this as a center, the first nearest neighbor is this particular atom. If you take this as a center, the first nearest neighbor will come with this atom, and these distances are actually measured and they plotted as a frequency versus the nearest neighbor distance. Fine, so you can see that the pink color one is the random, but the red color one is the experimental one. And you can see that these red color atoms, the tin atoms, it evolves as a highest frequency. The frequency increases at this particular nearest neighbor distance, which might be around 0.3 nanometer. So most of the atoms are at a distance of nearest to the 0.3 nanometers. Okay, and the frequency is very high. Okay, so and if you see that as the nearest neighbor distances increases, you can see the distribution also becomes less. Okay, and you will have a hump here. It means that these number of atoms, which is of 200 frequency, this number of atoms has a distribution distance between 1 and 3, the nearest neighbor distance between 1 and 3. Okay, so this is how we can calculate the first nearest neighbor distribution. and this distribution distance is nothing but these are compared with the random distribution which is done by the binomial distribution function. It means that binomial distribution function, it means that the atoms are randomly distributed in a solid solution. Okay, so this particular example, it is talks about the copper atom. So here this is a tin atom, here you can see the copper atoms. Okay, so it is the copper atoms with the random and copper atoms with the experimental first nearest neighbor. Okay, so each atom, the nearest neighbor is calculated. So for this particular, the nearest neighbor will be this, for this particular nearest neighbor will be this. Okay, so this is, and you can see that this is the random and this is your experimental part. You can see that it is shifted towards the left. It means that there is a tendency of segregation, tendency of clustering of these copper atoms. Similarly, for the tin also it's similar. So it means that the tin atoms actually in the solid in this are not randomly arranged. They have they are clustering together, they are coming together at certain locations, or we can call it as they are aggregated in the solid solution. Okay, so that's why there is a shift in the peak. There is a shift in the peak, either for the copper or either for the tin. Okay, and you can see that as I told you, this tin, this particular hump, it means that very less number of tin atoms are far apart, of approximately of sizes between 2 to 3 nanometers, and these numbers are very less. However, the numbers are very high, of the frequency of about more than 600, where their distance between the tin atoms are 0.3 nanometers. Okay, and this distribution not only for the first nearest neighbor, it can also be measured, it can also be plotted with the fifth nearest neighbor, 10th nearest neighbor, or 100th nearest neighbor. Okay, so depending upon what type of nearest neighbor atoms you want, you can actually plot by first, fifth and 10th and 100th nearest neighbor. Okay, so we call it as a kth nearest neighbor distribution. It means that examination of interatomic distances between each atom to its second nearest neighbor, or the third nearest neighbor, or the kth nearest neighbor atom. Okay, so here I will show you the distribution of copper for the fifth nearest neighbor distribution as compared to the fifth nearest neighbor binomial random distribution. Fifth nearest neighbor means, if you see of this particular copper atom, blue color, you can see that the closest is 1, second closest is 2, third closest is 3, this is the fourth, and this is the fifth. So the distance between the first, the zero, the origin atom, and the fifth atom, that particular distance is measured and is being plotted in the frequency versus KNN nearest neighbor distance. Similar analysis will be done for each atom. Okay, so this is the first, second, then you will have a third, it might be fourth, and this might be the fifth. So this particular distance is measured and plotted as a frequency between the KNN nearest neighbor distances. Similarly, you can actually plot the 10th nearest neighbor distribution. Okay. Okay, so with this, now the next is the cluster identification. Okay, now we will talk about, we have talked about the frequency distribution, and you can see that how these are compared with the random binomial distribution, and it shows that these some of the atoms are aggregated together or segregated together. Okay, so based on this, from the atom probe reconstruction, what we can do is cluster analysis. And this cluster analysis is done by several algorithms which are based on certain assumptions. Okay, so what is the concept of cluster analysis? As name suggests, these are the clusters. Okay, it means that identification of clusters or the agglomerated atoms from in the data set, and it can be done by using a certain assumptions. Okay, so if there are two or more atoms are associated or are nearby with one another, then you can use several algorithms which can differentiate between the clusters, or which can differentiate between the precipitates or phases. Okay, so based on the algorithm or certain assumptions. So the most common algorithm which is used is the maximum separation distance. This is the most common assumption or most common algorithm which is used to identify the clusters in the data set. What is the maximum separation algorithm? It is based on the assumption that the distance between the two solute atoms, that are clustered together is smaller than the distance between the two solute atoms which are found anywhere in the specimen. Smaller than the distance between the two solute atoms which are found anywhere in the data set. So there is a term which is called D maximum. Okay, so if the distance between the two solutes which is present in the data set is less than the D max, then these are termed as a cluster of these atoms. Okay, so it means that the algorithm, what it will do is it will group together the atoms that are separated by less than the distance between the D max, less than the D max. That is, if there is a solute which is P and Q are separated by a distance dp and dpq, then the cluster algorithm will find out the clusters which are having a dpq less than the D max. These are identified as a cluster, one cluster. Okay, so it means that here the P and Q are considered in the same cluster. Okay, so what is the parameter which controls in this assumption or in this algorithm? The parameter which is controlled is D max. Okay, so with this parameter what we can do is we can filter the clustered from non-clustered atoms on the basis of maybe first nearest neighbor. Okay, so here I am showing you an example. Okay, here you can see that these these are different kinds of atoms which are gray and blue. Okay, and you can see that if you find if you assume that D max is chosen for the maximum distance, separation distance between the two solutes or between the two atoms, then you can see that if you point out this particular atom, and if you draw a distance of D max and a circular or a sphere around this particular atom, the whatever the atoms which is having a distance less than the D maximum, then these atoms are together called a one cluster. Similarly this particular region, okay. So you can actually draw several clusters with this assumption that there is a D max. Okay, so a sphere of D max is drawn around each atom of the solute. If another solute atom occurs to be in between this D max, if it is present in between this D max, then these we call it as a one cluster. Okay, so this is a one assumption, and this process is repeated for each solute atom. This process is repeated for each solute atom. Finally the clusters are assembled by connecting the atoms together via a network of common associations. Okay, so you can see in the C, for each atom if you calculate, then you will have a cluster of these common associations. Okay, so these are termed as one cluster. Okay, but you know that in atom probe we have a local change in density, atomic density, or also the APT resolution are dependent upon the trajectory aberrations, local magnification effects that might directly provide the error in the results or identification of the clusters. Okay, so this particular is based on the D max, the distance. So this analysis is based on the D max, which is the distance, correct? Now we can also identify the clusters based on the composition or the concentration. Okay, so here and this so we can also do the cluster identification based on the iso based, means composition based. Okay, and here instead of distance solute concentration or composition is used as a parameter to define the cluster. Okay, so a radius, so usually what it has done is similar to these atoms which are present, a radius of D max is prepared in the earlier method, but in this case a sphere of radius R max is constructed around each solute atom, and the concentration CS of solute atoms inside the volume of that particular R max is measured. Okay, if the concentration which is measured if the concentration measured is greater than CS minimum, then the atom is considered as a clustered atom. Okay, so this is another method where you can actually use the solute composition as a parameter to define the cluster. Okay, so so the solutes and this R max, the distance, the R max for each solutum is plotted such as there will be a sphere, and inside this sphere you will have a some solute concentration. If that particular solute concentration is greater than this CS minimum, then we can tell it as these solute atoms belongs to the cluster atom. Okay, so this is one thing. Another thing is the radial distribution function. The third thing is radial distribution function. Okay, well, we call it as a RDF analysis. Okay, RDF analysis is used to identify the structure in a material by examining the average local neighborhood as a function of distance extended radially from each atom in the data set. Okay, so RDF usually builds the description of local atomic neighborhood and provides average. Usually it provides the average radial concentration profiles starting from each and every detected atom of chosen element of solute or the chosen atom. It actually provides the average radial concentration profiles. Okay, so for example, if there is a probability of the probability density of finding an atom J at a radius of x, y and z coordinate. Okay, so when an atom I is at the origin, then the RDF is calculated as CER divided by C naught, which is equals to NER divided by NE NR divided by C0. Okay, so what is these terms? These terms are nothing but CER is the atomic composition of element E at a distance R. Atomic composition of element E at a distance R, and C0 is the average composition of E in the analyzed volume. Similarly, NR is the total number of atoms at a distance R from the solute or the selected or the chosen atom, and NER is the total number of E atoms of that particular element atoms at a distance R. Okay, so this is the third way of doing identifying the cluster analysis, which is called a radial distribution function. Okay, and this term radial distribution function and the cluster identification by the solute concentration and also the cluster identification based on the maximum separation distance. These are the three basic methods to identify the cluster of any tomography data. Okay, so in after few classes, what we do, I will also cover some standard data sets, and I will show you how these clusters is identified through these algorithms in the standard software. Okay, so with this I will end this class now, and we will continue with this topic with certain other distribution maps which are very important to understand the crystallography or the interatomic spacings, correct? And also we will cover the effect of detector efficiency on these cluster analysis. We'll just basically cover these topics. Okay, thank you.